right, it is 9 o'clock in the morning here on the West Coast, so it is time to get started. Hello, everyone. Good morning, good afternoon, or good evening, depending on where you're calling in from. Thank you for joining us today for the live training that we are going to present on our CI 110 Plant Canopy Imager. A little light housekeeping before we get started. Please, if you have any questions about the content of what I'm showing you, as far as questions about operations of the instrument, this is going to be very much a walkthrough of the instrument. So if you have questions about what I'm doing, how these instruments are being used, feel free to put that question in the Q&A. If you have just a comment, you are just commenting to other people that are attending, please leave that to the chat section only. Also in the chat section, please be monitoring that because our moderator today is our marketing manager, Michael Williams, and he is going to be posting any sort of relevant links that you might want to click on. Those will be posted directly in that chat function, so please check that out. And leave the Q&A only for questions that are relevant to this presentation. Thank you. Also, if you're experiencing technical difficulties, if my video freezes up, my audio freezes up, please put something in the chat for that so that we can address that while this training session is occurring. A little bit about myself. My name is Galen. I am the Director of Applied Science here at CID Bioscience. I've been with the company for 3 years now. I have a bachelor's degree in biochemistry and a master's degree in food science from Michigan State University. I'm an IFT certified food scientist, and I have a lot of experience in food quality testing and safety assessment in the food, agriculture, and cannabis sectors. So today's agenda, we're really just going to do a little bit of light discussion about who is using the CID 110 Plant Canopy Imager, how the actual instrument works. We're not going to get too deep into the mathematics of it. There's a lot that we could get into, but I'll provide some resources for you to learn more about that. And then we're going to get right into unboxing the instrument. So actually showing you what it looks like when you receive the instrument, what all the components are, explain all the different parts of the instrument, the hardware parts. And then we're going to get started on the software aspect of the instrument, take you through all the menus, what all those do, show you an example measurement. And then I'm going to actually show you how to access that data on your computer and how to work with that. And then at the end, we'll kind of go over some troubleshooting stuff. You know, if you need to update the instrument, if you also need to get help support, we can show you those resources on our website. So the 110 is a hemispherical photography instrument. It is used by ecologists and people in the forestry industry, as well as people in commercial agriculture, to essentially help monitor plant health, biomass production, things of that nature. And so it's a pretty versatile instrument. And as I just mentioned, it utilizes a technology that's called hemispherical photography. And it uses this as an indirect way to measure LAI, or leaf area index. And now the theory behind this is essentially we're taking a photograph, and it's a hemispherical photograph from the ground up of the canopy. And we're taking that photo and we're turning it into a grayscale image. And that grayscale image then has varying levels or degrees of grayness throughout that entire image. And there's a method, what we call the gap fraction method. And what it's doing is assigning a threshold value on that grayscale image, on that histogram of grayness, those different varying levels of grayness. It's establishing a threshold value that will determine what is sky and what is plant. And so we say leaf area index, and what we really mean, because this isn't distinguishing leaves from branches, from tree trunks, it's really actually more of a plant area index. And so there are ways, and I can show you ways to help make sure that you are only getting leaves if you really, if that's what you're most interested in, but just beware of that kind of distinction. And so that's essentially how the instrument works. It's just a camera that's on a auto leveling gyroscope, and it will take those images, run those through our software, which does that gap fraction analysis where it establishes a threshold, and then it establishes what is the sky versus what is the plant. So what I'm going to do now is actually transition over to our unboxing, and I will stop sharing my screen and change over my cameras so that we can actually get into seeing what all comes in a package when you get a CI 110 instrument. So this is what the CI 110 case looks like. It's just a long rectangle. We got 3 different latches here. I'm going to go ahead and open the case itself. And everything's neatly kind of organized inside of this case. It's all got a foam insert. It's very protected. First thing you'll notice is the instrument itself. It's the long elongated rod right here with the actual screen. We have a manual, an actual printed hard manual that comes with it. So in this manual, you can find lots of good information about the theory, as well as actual menu structures, what the menu structures, what each menu does, as well as actual calculations that we utilize in this device, and some troubleshooting tips, battery replacement, and all that kind of information, all located in the manual. We also have a battery charger. So we have the plug for the battery charger, as well as the actual battery charger itself. So this instrument uses 3 rechargeable lithium-ion batteries. So we have a charger port here that is easy for charging. So when you're in the field and you need to just replace your batteries, you can just easily pop in those rechargeable lithium ions. In this pouch here, we've got a lens cloth that's just for cleaning off your camera and your screen. We also have here 2 neutral density filters. And so this is a feature that I'll explain to you here in a little bit once I pull the instrument out. But we include 2 of these at different optical densities inside of this as well. Here we have our spare battery set. So it comes with a set of batteries, and then you also have a spare 3 batteries that comes with it. We also have a convenient carrying strap. So this is actually a strap that you can use as like a sling to carry the instrument around. We also have keys so you can lock your case. Each of these latches have a lock, so you can easily keep it under lock and key if you need to. Now let me pull the actual instrument out and show you some features of the instrument as far as the hardware is concerned. What we have here is first the actual camera. So as you can see, when I tilt this back and forth, it is a auto leveling camera. So it's on a gyroscope. No matter what angle I kind of hold this at, it's going to stay leveled. And as I mentioned about the filters, the nice thing is that this is very easily compatible with any kind of camera filter, physical camera filter, where you can just magnetically pop this off, and then you can put your filter right in there, and then this just latches right back on. It's magnetic, so it's very convenient to put your filters in and out. Along this arm here, you can see it's all white on the top here. This is all lined with PAR sensors. There's 24 different PAR sensors in here. And so those are going to be measuring the photosynthetically active radiation from 400 to 700 nanometers. Here we have the actual screen itself. This is a touch screen. We have 2 different ways of navigating the screen. You can either touch screen or you can use the keypad down here, the 4 arrows. There's also the power button right there. How you actually take the measurements, if I turn this on its side, there's a trigger right here. And so the trigger is what you actually use to take the measurements. So when you hold it, you get a live image. When you release it, it actually acquires that image. If I flip this upside down, you'll notice right here, there's a USB port. So this is actually where your data is stored. So you can store it locally with that USB. We also do have a Wi-Fi option. So if you do want to transfer data wirelessly, you can do that as well. This is just an armrest. So if I were to hold this, this kind of rests along my arm, helps with counterbalancing the weight, makes it a very convenient instrument to use out in the field. And then the last feature is right here is the battery cap. If I unplug that, by twisting it, you can see the batteries just go into this battery tube there. So that is the instrument hardware. It's a very simple, elegant instrument, very easy to use, very lightweight, portable out in the field. And so what we want to do now is actually dive into what it looks like when I take a measurement and all the actual software options within this device. So I will once again switch over my camera. We'll get a new view here. So this is what the screen looks like when I power the instrument on by clicking the power button, and I have no measurements stored currently in my device. So my USB drive is empty. There's no device, no images stored currently. It's a completely empty device. It is prompting you to do one of two things. You can either press and hold the trigger to record a new measurement, or you can press the left arrow to access the main menu. Now, as I mentioned, this is also a touch screen. So if I wanted to access the main menu, I could click this left arrow, but I could also click right here. And so this actually right there, that little back arrow takes me to this main menu as well. So in this main menu, there's a number of different options that we can choose. The first is the file name prefix. And so this is your first method of organizing your data. And how this works is if I click the right arrow, or if I just click it on the screen, I can change what the prefix is of my measurement. And so every measurement you take with this prefix, you can take 20 in a row and it'll name it demo _ 0 through 20. So it automatically will apply a numerical value after an underscore after the prefix. So you can name this after a specific, say you're doing orchard research, you can name it after a specific orchard or a row or an area within your orchard. You can, if you have already established names, this prefix can contain both letters and numbers. So when you enter it, you just click the up and down arrows to cycle through your letters. So I can cycle through that way. Click the right arrow to progress. Left arrow deletes. So if I want to add that O, I just go in there and add that in by clicking the right arrow. Click the right arrow again to go all the way to the end. And now if I want to save this, I click the right arrow one more time. And it explains all that as well down in that menu as you saw. My second way of organizing data is by changing the data folder. And so if I click the right arrow from data folder, so if I click the left arrow, I will navigate back. If I click the right arrow from data folder, it goes into that menu. I click the right arrow for USB disk. I can create a new folder or select a current folder. So if I wanted to create a new folder and call it demo, I could do that. And then, or I could select current folder. So that is our two main methods of essentially organizing your data. You could have both prefixes and different folders to help you organize data collected out in the field, so that you don't have to worry about doing all that in a post process. The export data feature, this is what we'll use to actually put the data either onto the USB drive or onto the network. The network feature, this allows you to connect to Wi-Fi. And so when you want to transfer data wirelessly, you can do that, or by doing the export data while you're connected. You can also, though, use this feature to update your instrument if we have software updates available. And I'll explain that later on when we're talking about the troubleshooting section. Under the advanced menu here, here's where a lot of the troubleshooting stuff kind of takes place. If you have an issue where you notice your image isn't centered, properly centered, like as if the camera was a little bit out of alignment, you would go into the camera alignment menu, and you could change your X and Y offsets to make sure that that camera is aligned so that it's pointing perfectly straight up. The sensor calibration, this is something you shouldn't have to do. We do all these calibrations and alignments in-house before we send you the instrument. However, if the need arises, this is how you would access the sensor calibration. This is for the PAR sensors that are along the line, lining the whole arm of the instrument. And so to do the zero calibration, that's a pretty straightforward process. You just have to eliminate all light that is hitting the PAR sensors. So if you just cover it up with a very, very dark light blocking blanket or something like that, you could do the 0. The span calibration, a little more difficult to do. We don't recommend users do that. If you do think that your PAR sensors are out of calibration, then please contact us through our support and we can figure out a way to get that calibrated for you. Compass calibration is a fairly easy process that the user can easily do if you think that your compass isn't aligning correctly. It's just a series of movements. You'll have to turn the instrument 360 degrees for pitch, roll, and yaw, so that you're doing it at all 3 axes, and that'll recalibrate the compass for you. Changing the system time, it's a pretty straightforward concept. This is just letting you know the version, software version number of the instrument. So if there is an update, you can compare whether you have the newest version of the software on your instrument or not. So if I click back on the left arrow, I go back to this, and I'm back to this main menu structure. I can exit the menu by either clicking the left arrow or by clicking right on exit menu. And once again, we're presented with our option. So we've already done the option of pressing the left arrow to show you the main menu. So now we're actually going to press and hold the trigger to record a new measurement. So my finger's down here. I'm going to hold the trigger down, and I'm going to get a live view here. And so you can see my hand, you can see the top of my head, you can see my face a little bit there. And so because we're in an indoor setting, there's actually no plant canopy. I'm just going to put my hand here to kind of pretend that there's some canopy here. And one feature that you should definitely know about is while I keep my hand over here, while you're holding the trigger down, the left arrow, if you press that, will auto expose. It'll change the exposure automatically. The up arrow will open the exposure. Down arrow decreases exposure. So up arrow increases, left arrow automatically. What I recommend, and this is coming from years and years of this technology being used, is that it's always better to err on the side of overexposure. So I would usually recommend that you auto expose and then maybe press the up arrow once. And then from there, I'm going to release the trigger. And you can see that the instrument already is trying to make some decisions about what is or is not plant canopy. Obviously, none of it is actually plant canopy, but we're kind of tricking the instrument into thinking that it is. Now, there's a lot going on this main screen here, and it can be a little overwhelming if you're not sure what's going on. So in the image itself here, the green is what the instrument is identifying as plant canopy. And the non-green is what it's identifying as sky. The orange graph here on the top is showing you for every single PAR sensor along the arm of the instrument, what they're reading for PAR levels. Now it'll take the average, it'll calculate the average for you right here. And PAR is in micromoles per meter squared second. And then we also have this SunFlex option. And so the SunFlex option, and we can talk about this here in a little bit when I talk about the next menu structure, is really determined by what the user wants to define it as far as what it is a parameter that is not necessarily direct sunlight, like an open sky in the canopy, but it's an incidence of light getting through in a short period of time, light getting through the canopy. And then over here in the green, we have the PAR LAI. So actually this instrument can calculate LAI using 2 different methods. The gap fraction, which is the main method that the hemispherical photography utilizes, but then also we have the PAR LAI, which is actually using the PAR sensors and the PAR value to then calculate LAI. The big number obviously is the gap fraction LAI. That's the main measurement. We have a canopy density. That's a calculation that's based off of the gap fraction LAI. And then we have our leaf angle, which is also another calculation based off of the LAI. And down here, you'll notice we have our date and timestamp. We have our location, our GPS coordinates, and we have our file name. So as I told you, our demo is our prefix, and so the 0 is the actual first measurement there. And so all of your data, when you export this, and you'll see this later on, all of your data is going to contain all this information. So every individual measurement, you'll be able to pinpoint to an exact spot where you took that measurement and exactly what time of day, what day, and all that. So all that information is traceable, and you'll be able to easily access that. From a measurement, there's a lot you can do here. This was just the initial measurement. Okay, sorry, we had a little technical difficulty there. So there's a lot that we can do with this image that can actually be done in post, I guess is what you could say as a term. So what we want to do is click the right arrow, or click this right arrow up here on the top of the screen. And we're going to go up to image. And so in this menu here, we can see the various ways of adjusting our image. And so the exposure was just kind of the first step that we can do while we're acquiring the image. There's a lot we can do with the image afterwards. We can also change all these things and then set them as the default. And so if I change the brightness here, what that's going to do is actually, when you increase the brightness, it will, if I click the right arrow here and I put my brightness up to 100, it's going to wash out completely. If I bring it all the way down, it's going to be a super dark image, completely black. So this is going to be something that the user kind of has to define for themselves and see what works best for their specific environment that they're doing their image acquisition in. The default is just set at 50. Same with contrast. So contrast will actually help differentiate your canopy versus your sky a little bit better. So, right, so here you can see our lines are a little bit more clear where this kind of yellow wall is, and where my hand and the tripod over here is. So as we were increasing the contrast, that actually might help. So if I want to save this setting, I'm going to click the right on 65, click the right arrow. And then if I click the left arrow to go back, it's actually going to apply those changes. And now if I go back to that image, we also have these red and blue and green filters. This is for varying conditions of sunlight. You might want to adjust and the degree of redness in your image. And so what this is doing is applying a filter, which means 100% red filter means no red light getting through the image. It's just blue and green, as you can see in that image. If I wanted to take out the blue light and just leave red and green, I could also do that. And these just help, or just remove green entirely. I could do that. Now it's just red and blue. And sometimes these help, sometimes with the varying lighting conditions. If I wanted to establish for a certain day that I'm acquiring images that these will be my defaults, I would go back with the left arrow. I would go down to save and set defaults. And what this is going to do is it's going to save whatever I did to this image. It's also going to save it so that any future image that I acquire, it's going to use those values, those filters that I put in the image. Those are going to be my new defaults. So that helps with consistency. If I go to this gap fraction LAI menu, then what we see is various inputs here for threshold, resolution, zenith divisions, and azimuth divisions. So this is a pretty important menu for determining the actual gap fraction LAI. The threshold specifically, as I mentioned, that's the threshold that is being used to determine what in the grayscale image, what is sky and what is plant. So if I am if I want to change the threshold and I'm wanting to use the Atsu method, then I can choose that. And that is a gap fraction method that's been studied previously and has been shown to be useful for low gap fraction. If you have low gap fraction, the Atsu method is a pretty good method to use. The entropy crossover method is another method that's been studied. These are just methods for establishing a threshold. They are mathematical, essentially algorithms that help determine what the threshold should be. And so we have this Atsu method, we have the entropy crossover. We also have a manual option. So if you want to decide what I think, what you think the actual threshold should be, the Atsu said it was 169. What if I wanted to try 145? I can apply that threshold. And now you can see that we're changing what it's determining as plant and what is skies. If I can even go a bit lower, save that. And now it's saying my hand's not even plant anymore, which was the whole point of me using my hand, was to try to mimic plant. I could also increase this past 169, and we could see at 200, now there's a lot of plant, or what it's identifying as plant. So that's how you change your threshold. This is probably the most important, along with the exposure and your contrast, the most important ways to manipulate what exactly is plant and what is not. A good troubleshooting tip is to go out into your canopy, take an image, and from that image, go through and manually identify with that image what you know for a fact is plant and what is not. You can also use this to manually identify if you are really trying to narrow down not getting any branches or tree trunks or other non-plant obstructions. If you want to try to not have those be part of your threshold so that they're identified as sky and not plant, you can also just basically take that image, go through and manually just kind of do some trial and error work to figure out what do I need to do to set this, what threshold do I need to set this at in order to make sure that it's consistently only identifying what I want it to identify as plant. The resolution, if I go back to this menu here, the resolution is always going to default to the highest resolution. We highly recommend you leave that as the highest default resolution. Unless you are really concerned about file size, then you can decrease your resolution, but this is an 8 megapixel camera. We highly recommend you keep the highest resolution possible. Zenith and azimuth divisions. This is an important one for kind of eliminating potential non-plant obstructions in your field of view from the picture. So actually, I've got a pretty good example here with my azimuth. If you guys aren't aware what zenith and azimuth are, zenith is kind of like, think of it as the angle of the sun is a really good example. So when the sun is changing its height in the sky, that's the sun's zenith angle. And so it is the axis that is actually coming out perpendicular from the screen. So if I had a pole sticking straight out from the screen up towards me, that is my zenith. And then every angle from the flat surface going up to that perpendicular, those are my zenith angles. And those are represented by these concentric circles on this image. And so every circle is a new zenith angle. The azimuth is the actual, what you're used to seeing on a flat 2D plane as seeing angles. So those are just the 360 degrees, every kind of like a clock. Those are those divisions. And what you can do here is customize how many divisions you want to separate your image into and what angles you want to start and end at. So if I wanted to change and say, I don't want to include, for example, maybe there is a bunch of noise on the outermost part of my image, and I don't want to include that. What I would do is go into my zenith angles, because I'm trying to eliminate a whole ring here essentially, and I would change my starting and ending angle. So my ending angle, my starting angle is 0, which means that I'm starting at the pole sticking straight up out from the center of the screen, and I'm ending at 75, which is where the last concentric circle ends. So if I wanted to change my ending angle to, say, 70, now you see we're eliminating that outer ring from the image. So say I wanted to eliminate even further to get rid of everything except for maybe my hand. I guess we kind of have to just see how much that's going to be, but let's start with 50, see how far we are. So that limits my image even further. Now, if I wanted to, you can also obviously customize your number of divisions. You can customize the starting and ending angle. But so if I actually wanted to eliminate, for example, the very center of the image, I could do that as well by changing my starting angle to 10. So now we're only getting this donut of an image. We're eliminating what's in the very center and what's on the very outside. If I wanted to change the actual angles that are being used, as far as the azimuth, we can also change that. We can say that we're not going to use anything from, say, 15. We're not going to use anything from 0 to 15. And we're not going to use anything beyond, let's say, 285. So we're getting rid of that whole section. So now the only part of the image we're actually analyzing is the stuff that's in white, not the grayed out part. So that's how you would filter out any sort of empty space. If you have big sections of empty space, or a tree trunk, or if you have yourself or another person in the image for some reason, or equipment or anything like that. If you need to get that out of the image because it's identifying as plant when it shouldn't be, then you can use this tool as well. Now the PAR LAI is something that requires a little bit of extra work, more than the hemispherical photography does. We do have some requirements in the calculation that need to be accounted for. Now, some of them we can do automatically, such as the solar zenith. When you take images with the CI 110, you should always be facing north when you take the image. North is identified as this red dot on the image. So when you're taking the image, you want that to be aligned with north, the top of your screen. So when you take an image, just for consistency's sake, you're taking it while you're facing north, and the GPS is actually then going to be able to calculate based on your location and the time of day and the day what your solar zenith angle is. So you don't have to manually input it. You can manually input it if you want, but you don't have to if you're using the instrument correctly. Same with your leaf angle. So if I click into solar zenith, you can change it manually, but it also is currently using it based on GPS. Leaf angle distribution, you can do multiple things with this. So the leaf angle distribution, you can enter it manually. If you know your leaf angle distribution within whatever species you're studying or within the forest canopy, then you know that leaf angle, then you can certainly type that value in. But if, as it says, to use the value calculated from gap fraction analysis, enter 0. So if I wanted to enter 0, I could just do that. So that it's using essentially using the calculated leaf angle based on the gap fraction calculation. The one thing that we can't calculate for that you need to do is take a measurement of light above canopy. So this provides the essentially the reference for the PAR LAI, the no canopy cover reference value. So you can either manually enter that by taking a measurement, with another light sensor if you already have one or anything like that, and you can just enter that into your light above canopy reading. And then that will allow us to get a more accurate LAI, par LAI. So then you can compare the par LAI to your gap fraction and see what kind of differences you're seeing just based on the two different methodologies. Sunflex, as I mentioned earlier, that was it's essentially a parameter that is determining short light incidences that happen in the canopy that aren't necessarily due to it being gaps in the canopy itself. Just movement, from wind or whatever, small incidences of sunflex. So what you can do here is actually enter a par threshold to use for sunflex. And so this is in micromole per meter squared second. So you can enter an actual threshold that you want to use to account for sunflex. As it mentions, you can enter 0 for automatic. So I'm going to enter 0 for automatic. But you can also set it manually, as I mentioned. A nice feature as well for these measurements is that you can rename them. So if you decided, or you forgot to change the prefix before your next round of measurements, and you're at a new field, you're in a new spot, you can go and actually change your prefix here. And so this is good for if you make a mistake. This is not the way you want to go about it as a proactive measure. This is more of a reactive measure. You don't want to have to do this every single measurement. So once you find out, oh, I need to rename this, then you would go back. I guess it's the saying already has that file. So we're just going to rename it demo 8. And now what I would do here is for future measurements, I would click the left arrow to go to the main menu. And then I would go to my file name prefix. And I would change that. So when I click this is a good demonstration of when you already have images on your instrument. This is how they look when you turn the instrument on. If I want to access this image, I click the right arrow and it takes me back to this actually analyzed image. So I'm going to click the right arrow again, just to go over our last few menu structures here. Save, so that's just saving the actual measurement. If you were to change all your settings like I just did. So now that we have this saved, now it's actually going to have all of these grayed out areas all saved. All my settings that I changed, those are all going to be saved. Save and set defaults is, as I mentioned, when you initially go out in the field, you're taking your initial measurements to kind of get your settings correct. That's once you get your settings correct, this is what you need to do. You need to save and set your defaults. And then, of course, we also have the option to delete our measurement. So we can easily do that just by clicking the right arrow here, and that will delete the measurement. So I'm going to go ahead and retake a couple measurements here just for fun. I'm going to take another one, just my finger. Okay, and so what I want to do now is move on to showing you how to get this data. Now that we've seen everything, that's all the menu structures on the instrument, how to actually go about changing all those. Now I want to talk about the actual data that you can get on your computer. So I'm going to switch to sharing my screen. We're going to pull the USB out of the instrument, but first we're going to shut off the instrument before we actually do that. And so now what I want to show you is, here's the USB. We're just going to pull this right out and we're going to put it into my computer and I will show you more data. And so my USB has been inserted into the computer. You can see here we have a USB drive. In here, as you recall, on the instrument, I created a folder that was called demo. And so now that data that I captured are going to be inside of this folder called demo. And each of those is going to have a name, demo 1 and demo 2. And so here is where we have our adjusted, measured, and raw PNG files. So these are all just images. This is the actual real image. Here is our grayscaled image. So this is what's showing you, and with our azimuth and zenith divisions. And here's our raw image. So this is actually the even wider angle. This is the image with no limitation on the actual on the outside there. So it's actually, this is like the 150 degree versus the 180 degree image. So same with this image with my finger, you have the same situation going on. My face looks really good in those. So if I exit out of this, let me see if I can close that. So what you can do with this data is actually, you know, put it into other softwares if you want, with other algorithms for gap fraction, other thresholding algorithms. You can easily do that. You can also post-process the images in some sort of photo editing software to even further increase the contrast between the sky and the canopy. There's a lot you can do in the post process here, because these are the actual image files. You can do a lot with different softwares. There is this very, you know, robust software on the instrument itself, but if you you know, want to do some things that maybe our instrument isn't capable of, or like I said, experiment with different gap fraction methods, you can always do that with the data here. So also, if I were, so I took those measurements, but I did not actually do any analysis on them. If I were to do the analysis, like if I were to actually save those measurements, it would have saved a CSV file for each of these as well. And that CSV will contain all the information as far as the calculations. And so it's just an Excel spreadsheet that has your PAR LAI, your gap fraction LAI, your sun flex, your canopy density, all your date and time, location, all of those calculations, leaf angle, those will all be in a CSV file from the instrument. Now, so that's how the data is accessed, and it's really simple and straightforward. There's no extra steps really needed. But I do want to show you guys for just general troubleshooting and just for further information for yourselves, how to access resources through our website, essentially. And so if you go to CID-inc.com, then click on the CI110 Plant Canopy Imager. We have just some general information here in these tabs. We have some theory of operation, light theory of operation, features, specifications, so you can actually see all the specs for the instrument itself. There is some information, extra information there. We also have, and this is very useful, so if you have a specific question about an application, and you're just kind of maybe curious about whether someone's used this instrument in a specific application, I highly recommend downloading our compilation of published research, because within that, you can get just a big zip folder full of research that has been published using our instrument. This technology, as I mentioned, has been used for quite a while as an indirect method for LAI assessment. So there's a lot of research out there with it. And so chances are someone's done something at least similar to what you've done before. Maybe not in a specific plant species, but certainly a lot of applications can be found within those published papers. So a very, very good resource there. Additionally, if you have a hard copy of the manual in the box, but if you need to access the manual online, you can easily do that by clicking the manuals link. And we have our instruction manual. We also have data and indice calculations. We have technical questions as well. So there are 3 different kind of documents that you can access to look at for more information about troubleshooting your instrument. We also do have a support and software link. So if I go to the support link, this is a very valuable resource with a lot of good FAQs So if you have a question about your instrument, a lot of them can be found here. And so there's information for like which gap fraction method should I use. There's really good paper out there that helps kind of describe what these methods are and what situations they're kind of best utilized in. How do I access the log file? How do I, what types of canopies? There's so many different questions that have been asked before. And so likely a question that you have is, very likely on this list of questions here. A very common one, the one I just clicked on, is the CI10 waterproof? CI110 is water resistant. However, if you're in a downfall, if you're in a rainforest during monsoon season, you're going to want to probably not have this instrument out and unprotected. So we don't recommend that you to use it in those situations. If you get it in like a very light Pacific Northwest kind of rain, then it might be fine. But we really don't recommend trying to utilize this without a waterproof case if you know you're going to be using it in a wet situation. And so this is great FAQs. As I mentioned, that was in the support link. You can also, as I mentioned, there is a software link. So when you go to update the software, you would, if we had a new version, which this is the current version is displayed here, you can download it here, and the tool, the actual tool is a wizard kind of tool, so it'll walk you through the steps needed to actually get your device updated. It simply requires connecting your instrument to the same wireless network as your computer, and then entering your IP address from your device into the software, and then it'll automatically upload that new software to your instrument. It's very convenient. A couple other things I want to mention. So actually I show you the manual. When you click on the manual, it takes you to a Word document link like this. Our calculations sheet actually shows you all the mathematics behind calculating all the different parameters needed for the leaf area index, transmission coefficients, talks about extinction coefficients, as well as mean foliage inclination. And then also additionally, we have, if I can pull this down and see it here. Here's the study that I mentioned about the actual comparison using our device of the different gap fraction methods. And so this is a really good paper to review if you aren't familiar with hemispherical photography to estimate LEI. This is a very good paper to start. There's also a really good review out there. All of that can be found in our published papers compilation. So highly recommend downloading that. Two more things. If you have specific applications and you've done your research, haven't been able to find anything necessarily in papers regarding how you want to use this instrument, you can easily schedule a consultation with either myself or our application scientist, Eric Munoz. It's a free 30-minute consultation to talk to us and pick our brains to see if this is something that application we think this instrument could be useful for, or other questions. So if you have application questions, please book that consultation. If you own an instrument currently and are in need of support, or in the future, if you need support for this instrument, you can click our support link and you can open a support ticket, and our support team will be with you in a very short period of time to help get that resolved. We try to be as attentive as possible, and we have an awesome support staff here. So don't be shy. Feel free, if you are in need of support, please use this feature. And lastly, if you are interested in the device and would like to get pricing, we do have a request pricing link. If you click that, you can simply just enter your information, and you can click on the checkbox for CI110 Plant Canopy Imager, and we will send you pricing information for that instrument. And that is essentially everything I needed to discuss with you about the instrument itself. Let me switch back over to our presentation. So we talked about unboxing the instrument, all the things that come with the instrument. We looked and dived into the actual features of the instrument, how to take measurement, the proper procedure for making sure you're aligned north, make sure that when you're holding that trigger down to get your live image, you're adjusting your exposure properly by using the left arrow for automatic, and then also using the up and down arrows for increasing or decreasing exposure. We talked about how you can remove the actual lens cap physically and put in physical filters. We include 2 neutral density filters. So if you want to keep that exposure really high, but you have really, really bright conditions, you can easily pop in a neutral density filter right into that camera, enter the optical density into the instrument, and you should be good to go. And so, as I mentioned, you can request a quote either through our website or through the tiny URL that Michael is going to be posting in the chat. So you can easily just click that link. It'll take you right to a page to actually get that quote. And if you are interested, you can follow us on social media, visit our website to stay updated on our current projects and new additions that are going to be coming to our product line. You can sign up for our newsletter. We have great newsletters that have a lot of good information on current developments in agricultural technology. Feel free to give us a call or visit our website if you have any other needs. With COVID going on still, we are not necessarily accepting visitors at this time, but in non-pandemic times, I would normally say, come on, if you're in the area, come on, stop by, and we're happy to have you at our office to give you a tour and talk about your applications. So thank you everyone so much. What we're gonna do is transition to the question and answers now, and I will do my best to answer your questions. We'll just get started. The first question is one of my favorites. It says hello, and I say, my answer is hello. The next one is from Mauricio, and he asks, hi, is this model similar to the former CI-110? So it is similar in the fact that it is also using hemispherical photography. It is drastically better in the sense that it is an all-in-one instrument. It's much more convenient to carry around. I don't have to have a tablet and a wand, so it's just an all-in-one instrument. Also, the camera has improved drastically since then, since the older version of the 110. So we have that, and all the software is improved as well, and it's the touchscreen interface and all that. So it's similar in the underlying technology of using hemispherical photography. It's just a much more refined and improved version. So the next question is, most of the new orchards are quite two-dimensional. How does it work for the calculations when the canopy is not three-dimensional? So we have had people use this in orchard applications with some success. And actually, I highly recommend something I forgot to mention earlier on is we have a great webinar that we did with a couple of users, 3 different users of the CI-110. One of them was a stone fruit orchard, I believe, using the instrument. And I highly recommend watching if you want to get some really good advice on best uses with this instrument in orchard applications. Highly recommend checking out that webinar. Michael, if you could post that link in the chat, that would be great. For the guests here, I highly recommend checking out that webinar. But yes, so it should work just fine, even though the canopy is not three-dimensional. I think it's just going to require some additional changes to your settings when you're actually doing the measurements. It just requires some customization on the end of changing things like your leaf angle distribution and stuff like that. One thing I will say is that gap fraction in general does assume a random distribution of leaves. And so that is kind of, once you start messing with assumptions that are made within the actual calculations of these methods, then that's when you can start seeing changes in accuracy. So that's just one thing I would keep in mind, but highly recommend checking out that webinar. Good for getting more in-depth information on uses of this instrument in orchard settings. The next question from Jack Ingle. Hi, Jack. Good to see you. Is there any charge for the software updates? The answer to that question is currently no. So no, when we update the software, we will have it on the website. We'll send out an announcement, and everyone will be able to just download that and update their instrument. So the next question is, why is the value so different between the gap fraction and LAI par? So a couple things. One, please don't take any of the measurements that were being taken to heart. We are not in a canopy. We are in an artificially lighted setting in an office. This is not in any way the intended use of this instrument. So those values are not by any means supposed to be accurate or representative of what is actually happening. But there are going to be differences in your gap fraction and LAI par likely because they are just two completely different methods. They're two different methods. And so they might align up in some ways, but they also differ in others. Gap fraction is using a completely different technology and underlying theory than the LAI par. So it's just providing you with extra information so you can have both and you can see, okay, well, this is what the LAI par is doing in this canopy over time or you know, over this distance of this row. And then this is what the gap fraction is doing. So you can actually, it's just really for added information. They're gonna likely differ. The next question is, what is the camera open angle? Thank you. So the camera is a 150 degree camera. Well, that is what it is when we adjust it. Technically, this is a 180 degree camera. But if you use 180 degrees, you're going to get the instrument, you're going to get yourself in the image. So it's just better to kind of limit it to more of a 150 degree kind of cone instead of letting it be a complete hemisphere. You can change that if you want. If you really want to have to, you know, see everything and put in the extra work to get yourself and all the other stuff out of the image, that's totally up to you, but we recommend keeping it at 150. The next question was, I think, posted in the chat, but the next question is, why 24 par sensors and not 1 or 48? Why did we arrive on this number? I was not part of the engineering team that developed it, but I can tell you it's likely because that's the length of this instrument fit 24 par sensors perfectly. And I'm guessing that's why we arrived on 24. And it's better to have more than less so we can average them because they're in different positions, right? So we have ones that are farther out. We have sensors that are farther out towards the head. We have sensors that are closer towards the base of the instrument. So we can average all those together to see what kind of the average par reading is, as opposed to what you would get from just one. The next question is, what is the length of just the par probe bar? And that's a great question. I actually don't know off the top of my head, and I'm pretty bad at estimating lengths, but I would say that's like a little bit over a foot long. I would say it's probably like, yeah, probably something like 14 inches. And it has 24 bar sensors, as I mentioned. Okay, the next question is, thank you for the presentation. You mentioned the zenith angle is automatically calculated based on time of day, GPS coordinates. I was wondering if the precision of the GPS sensor in the instrument is high enough. Some GPS sensors give readings several miles from the true point. No, so our GPS sensor is very highly accurate, but if you are concerned about its accuracy, you can always reference it against another GPS that you are using. And you can always also type in manually, as I mentioned, type in the zenith angle of the sun for the LAI calculation if that is something that you need to, if you don't think you can rely on the GPS on the instrument. However, it is a very, very precise GPS system within the device, so I don't think you should have that issue. It definitely is not bad enough to be miles away from the true point, so you should be fine. The next question from Nyan is, Sunfleck is always coming in as 100%. Probable reasons? Probable reasons is because there is a constant light being shown on this instrument here, and so it's identifying that as par. It's coming in. It's Sunflex. Also, we didn't have the instrument set up necessarily to. I could have set it to limit the threshold of what we identify as Sunflex, and I could have made it 0 if I wanted to, but I didn't have that. I didn't alter that setting. As I mentioned, we're in the opposite of the use case of this instrument. We're in a artificially lit office space here with no actual plant canopy anywhere. So it's we're kind of just showing a proof of concept, not really getting accurate data here. The next question is there any difference between editing photos directly on 110 or software on PC? There's going to be a difference in the user interface. I mean, editing photos is something that can be done. There are lots of softwares out there that do this kind of image analysis for hemispherical photographs for canopy analysis. There is going to be a difference. You can, however, use this software on the PC as well. So if you needed to do the same stuff that you're doing on the instrument on the PC, that is something that you could do as well. So if it's easier for you to see it on a bigger screen and not use the touch screen and the arrows, that's something you could do. But the difference is going to be with the software. So there's always going to be differences. If you're using a different software, there will be differences. The next question is how do we make sunfleck measurements? So sunfleck measurements are happening automatically. It is, as I mentioned, that threshold setting with the par value. So what you're doing is you're saying in a certain period of time, if I'm getting this level of par for a shorter period of time, then what I'm saying is that is now going to be a sunfleck. And so that's something that you kind of have to define in the instrument and make sure that you're getting an accurate measurement. But it's calculated automatically when you take a measurement. And then the next question is the difference between par LAI and gap fraction LAI. I already kind of hit on this subject, but they're just two different underlying technologies. So the gap fraction is actually using the hemispherical image, and it's making a histogram based on the grayness and the image, and it's establishing a threshold to say what is sky and what grayness is sky, what grayness level is plant. Par LAI is actually using the photon flux, is measuring the photon flux and using a whole separate calculation to calculate the LAI based on that, as well as other parameters like the sun angle, sun zenith angle, and the leaf area distribution. So that is the difference. Next question is, how does 110 compare to the other non-image based LAI devices which calculate light interception from above and below canopy at different zenith angles? In terms of accuracy, post-measurement data processing, ease of use. This is a great question. So there's a couple different technologies out there that are used for canopy assessment, and what the advantages of hemispherical photography are just the amount of data that you're getting, and the fact that with the actual gap fraction LAI, you don't need to do the above and below canopy. With the par measurements and with light sensor based technology, you have to make sure you're getting on that same day an accurate representation of the light conditions. And so you either have to have a separate sensor at all times in a that's logging that data in a separate field that is monitoring, that is at least close enough by to have relatively the same lighting conditions. So that can get pretty messy. And also those instruments tend to have, they're multi-hand kind of operations. With this instrument, it's all in one. Hemispherical photography has been used for quite a long time, has a lot of research behind it, a lot of use cases, a lot of data to defend its use. And so it doesn't require as much user expertise and input. It's a lot easier. And in terms of accuracy, they're both calculating, gap fraction LAI. the biggest thing is with hemispherical photography is doing your best to remain consistent. So you have to, as I mentioned, you're always facing north when you take your images. You're always, applying the same filters to everything. So that is kind of the main differences there. With the post-measurement data processing, it's actually kind of nice with hemispherical photography to have all of your raw data information and be able to, if need be, have access to that. It's all about data transparency, as opposed to an instrument that just provides a readout of results, and you just have to just trust it and take it for its word. So there is that, it might be a little extra work, but you have access to all that raw data. So that is also like a very big advantage of the hemispherical photography. Next question from Derek. So Derek actually was one of the presenters on the webinar that I mentioned earlier. So good to see you, Derek. He's asking, is there a way to update the light above canopy for all images after they have been taken at one time, rather than updating each image individually? For example, I take 50 images and realize the light above canopy was not saved properly at the beginning. Can I update all these at one time? That's a great question, Derek, because that is a little bit of a bummer if you forget to save the default and then you need to go back and you've got a whole bunch of images you got to update. As far as I know, no, there's no way to do it all at once. You do have to go back and do each one individually. But I can talk to, I'll talk to our engineers and see if there is a workaround for that for you, so that you don't have to necessarily take the time if you forget to save those defaults. That's a pretty big punishment for forgetting to save the default. So yeah, thanks for asking that though. Next question is, how is the fisheye distortion corrected in the image analysis? Is a different ratio used for each zenith angle? That is a great question for an engineer. So I would very much recommend that you send that as an email through our support page, and we can actually have our engineers that design the instrument, that are optics experts, help answer that question for you. The next question is about gap fraction, zenith division 17 to 30 are coming as 0. Any reason why? I don't know if I truly understand that question. Nyan, if you want to send me, I don't know if I understand what you're asking. So if you can send me that email through our support page, I can help answer that question that you're asking. The next is, how is the data from PAR sensors used in image analysis? So it's not used in the image itself, it's used in the other calculations. So it's kind of like two supplementary measurements for the hemispherical photography. So the main instrument is the actual camera, the hemispherical photography, but then we added a whole secondary set of analysis by incorporating PAR sensors and allowing for additional measurements to be taken. So it's just adding more data to the other information you can get from the image analysis. So the PAR sensors are just totally separate. They're not actually affecting the image analysis in any way. They're doing their own calculations, like the PAR LAI, the Sunflex, the all the PAR for the individual sensors. So that's the average PAR that you're getting through the canopy. So that's what the PAR sensors are doing. The next question is it necessary to make measurements in diffuse light conditions, as is the case with the Lycor PCA 2200, or measurements under full sun can be done? So the full sun is tricky for all of these indirect technologies. So direct sunlight can be a little bit of a difficulty, but I have a couple of solutions. One would be that if you're doing canopy images in direct sunlight, if you can find a shadow to actually have just the camera part, be under, then that could help alleviate the direct sunlight issue. Also, this is going to kind of loop into the next question as well. The neutral density filters, that's the word I'm looking for, that we include are also good for this this purpose. And so just like with any other normal photography, if you have way too bright of direct light, you can use neutral density filters to help eliminate the potential, oversaturation of light by using these neutral density filters, the physical filter that goes into the actual camera itself. And so that's for the next question. I was asking about when do we use these. In order to have your exposure be high and in those direct sunlight conditions, you can use these neutral density filters to help alleviate that problem. So those are two possible workarounds for direct sunlight. But all of the technologies, have some difficulties. It is ideal, obviously, to do it in diffuse light situations, but that's not how weather works. It's not always diffuse light. So we try to make sure that everyone has options to use these instruments and in all lighting conditions. So yeah, well, thank you all so much. That's the end of our question and answer session. And I appreciate everyone that joined us today. I hope this helped clarify the operations of the instrument, exactly how it works. And if you need more information, as I mentioned, you can visit our website. This video presentation has been recorded, so we will send this out as a recording to all participants. And also, if you are interested in talking about your application, feel free to set up a consultation. And I think that I hope that everyone's doing well in the times of this world, and I hope that everyone's keeping safe and that you are able to continue on with your canopy research. And if you have been doing research with your instrument and it's something that you are wanting to showcase or share, we're always happy to promote our users' research. And so feel free to reach out to us if you have done something interesting or unique with your instrument. We're always happy to hear about how our instruments have been used. So thank you again, everyone. I hope you have a great day, and we'll see you soon.